This girl is great.
Shall we start the session, ma'am? Beatrice, ma'am, are you there?
Jyoti, ask Dr. Ghosh if he can start and just tell Kalidra then.
Yes, yes. Dr. Ghosh, shall we start the session?
Sure, go ahead.
Yes. Yeah. Calreader, you can start, ma'am.
Yes, ma'am. Welcome back to the lecture workshop on the OMIC sciences in the times of COVID pandemic. We hope you had a hearty meal, and now we will resume with the afternoon sessions. I hand over the platform to Sumeru, student of third year BSc.
Thank you, Kalvida. Be humble, be hungry, and always be the hardest worker in the room, said Dwayne "The Rock" Johnson. There can be no better words of wisdom than these for students aiming to reach the top. Good afternoon to one and all. I am Sumeru and I will be your host for this session. I hope that all of you had a hearty meal and are all set to listen and to watch out for this ocular topic. We have with us today an ardent researcher who is the director of GROW Research Laboratory, Dr. Arkasubhra. The GROW Research Laboratory focuses on understanding the genetic and molecular signaling mechanisms driving pathogenesis of ocular disorders and cancers. The gene therapy platform being developed here focuses on recombinant adeno-associated virus as the transfer vehicle, utilizing multiple pseudotype capsids for efficient delivery for targeted treatment of inherited diseases. A clinical-grade gene therapy vector production platform has now been developed here, and various functional genetic studies are being done with the aim of performing human clinical gene therapy trials in India. Apart from this, Doctor is also an adjunct principal scientist at Singapore Eye Research Institute, member of the National Committee for Gene Therapy, India, and a member of ICMR Research Task Force on Gene Therapy and Translational Immunology for COVID. Dr. Ghosh is an avid researcher and has co-authored 85 research papers, some of which have appeared in journals of international repute such as Nature, Nature Communications, Nature Genetics, and Nature Biotechnology. He has an amazing total of 9 patents and has been awarded the coveted Excellence in Research Award by the American Society of Gene Therapy in 2008. Thank you for being with us here, sir. Now, Dr. Ghosh will deliver his talk on the microbiome of the eye. We will have a Q&A session after the talk. I request all the participants type out their questions in the chat box. They will be answered by sir after his talk. So, over to you, sir.
Great. Thank you, Sumaru. And thank you, everyone, for being here. Thanks for the invitation to me to give this talk. Let me just share. Right. Can you see my screen?
Yes, sir.
Great. Great. So, good afternoon, everyone. So, as Sumaru has already introduced, I work on a variety of conditions related to the eye. And we are located here in Bangalore at the Narayana Health City, which is in Bomsamra. And in particular, I work for the foundation under Narayana Netralaya, the eye hospital. So it is very, very important to see, right? Everyone can agree. So it's the most important of our 5 senses. It allows us to see and interact with our environment. And this is a very, very complex organ. If you look at it evolutionarily, the eye is probably the most evolved organ amongst all the other organs and tissue types that we have. So primarily you can separate out the eye into 2 parts, the front of the eye and the back of the eye. The front part has what we call the cornea, and that continues on to what is called the sclera. And on the back of the eye, we have the retina. So the light passes through your cornea, through your lens. The lens, depending on the ambient light, helps you focus the image on the back of the eye and the retina, which then forms the image based on the visual cycle that ensues when light hits it. And that sends a set of signals through the optic nerve to your brain. This is how you see. And all of this is happening in nanosecond and femtosecond scales. So apart from this, there are obviously other parts, adjunct parts, if you would, which help keep the eye in its shape, in its place. So those are your eyelids, and then you have the lacrimal glands, which produce your tear films. And these are all the anatomical features of the eye. So there are a bunch of muscles and nerves, etc., that control the eye and support the eye and its movements. So the eye has always been thought to be an immune privileged organ. And just like the eye, there are a bunch of other immune privileged organs in the body. And these are your brain, your uterus, ovary, testis, hair follicles, etc. But amongst these, the eye is probably the most complex. Hold on. Just a second, it seems to have stalled. There we go. So, the ocular immune privilege, why is it important? It's because your vision really depends on a number of these tissues working in tandem and working in cohesion with each other to produce the clean image that you will see. So if you have inflammation, if this visual axis somehow gets distorted, then you'll not be able to see. So you can have blurriness, you can have complete loss of vision also. So each of these layers that the light passes through, starting right from the tear film that's on top of your corneal layers, across the different corneal layers, the aqueous humor, the lens, the vitreous humor, and right back to the retinal layers, are involved in this complex process of seeing. So therefore, the eye has a system of gates which prevent immune cells, infectious agents, etc. from getting into the eye. And these are the blood-retinal barrier, the blood-aqueous barrier, and the ocular surface barrier. So the ocular surface barrier is self-explanatory. It's the front of your eye. And this is the part that actually gets injured or has to defend itself the maximum amount of time, because this is essentially your UI, right? Your interface to the world. The blood-retinal barrier refers to the back of the eye. So the retina here, this needs to be fed because it's obviously one of the most highest energy consuming tissues in the body. So it needs a lot of blood and oxygen to be percolating into it. So that is fed by a complex system of retinal arteries, and these are lined with systems or gates which prevent immune cells from freely trafficking into the retina. And then you have the blood-aqueous barrier, which happens here. So the front of the eye, between the cornea and the lens, you have the aqueous chamber, which is filled with a clear fluid called aqueous humor. So what happens in disease is that this immune privilege sees a breakdown, or rather the immune privilege becomes restrictive. So that happens when you have injuries, infections, inflammation, disorders, etc. So this can happen in a number of different ways. First is the breach of the barrier, a physical breach. Suppose you get a tear in your cornea, or you had an accident and there is trauma to the back of the eye. So that can cause or provide an opportunity for immune cells or for pathogens to enter your eye, and that causes the immune privilege breakdown. The second is the loss of immune suppression. So inside the chambers of the eye, there are active systems that prevent the immune system to flare up inside. It prevents immune cells to dive in and accumulate inside your eye, because that will definitely cause loss of vision. And these immune systems, they respond to gradients of inflammatory factors. So these inflammatory factors are what call immune systems in. So you have to dampen that signal for immune systems also. So this immune skewing or dampening, which is a very active system inside the eye, that if that is somehow because of disease or because of trauma or because of an infection altered, then you can have a breakdown of your immune privilege. So why we are talking about this in case of microbiome will be evident soon. So if you look at the various kinds of eye diseases, the ocular conditions, so you have the conditions for the front of the eye, so corneal dystrophies and keratoconus. You have uveitis. So this can happen in the front of the eye, in the back of the eye, in the entire eye. And this can be caused by sustained chronic inflammatory immune response that happens due to bacterial infections, viral infections, trauma, injury, etc. Conjunctivitis, this is the one that you would all be quite well aware of. Most of you would have gotten it, pink eye. Then you get cataract. You can get glaucoma, which is a disease where your field of vision is marred because of damage to your optic nerve. You can get tumors, which is retinoblastoma, which happens only in children. So it's the commonest pediatric tumor. You can get genetic diseases like retinitis pigmentosa. You can get diabetic retinopathy, which happens as a consequence of diabetes, where people go blind. You can get dry eye disease, which is one of the most common diseases in the eye, where you feel irritation and bloodiness and all that. Computer vision syndrome, etc. So those are all related to dry eye disease. You can get AMD, age-related macular degeneration, where you get only peripheral vision. Your center of your vision becomes dark. And of course, ocular surface infections, because the ocular surface is exposed to the environment, so that gets the most number of infections. So what are the common causes of ocular infections? So you have a variety of bugs here, staph, pseudomonas, viruses like herpes simplex, adenovirus, enterovirus. And these can cause infections in the front of the eye, in the cornea, or in the conjunctiva. Or it can cause inside the eye, at which point if it becomes very, very fulminant infection, like an endophthalmitis, you might have to eviscerate the eye completely, because it cannot be controlled by antibiotics. So it's a terrible thing, but it does happen fairly often, and we see quite a few cases of those. Now, this is a data set from the Indian Journal of Ophthalmology, which shows you all the different kinds of infections that happen in the eyes. So you can have infections of the eyelids. So blepharitis is the most common, where your eyelid becomes itchy. You can have infections of the conjunctiva, infections of the orbit, the lacrimal gland and the lacrimal apparatus, infections of the cornea, and this is where you can lose vision very quickly, because if the cornea becomes translucent or opaque, the light can't pass through, and that is one of the highest causes of preventable blindness anywhere in the world, including our country. Infections of the sclera, infections of the various intraocular tissues.
Excuse me.
All right. So in order to fight infections, we need our immune system. So the primary functions of immune systems that are present in all tissues are to eliminate microbes. Yeah.
Could you please minimize your the window where it shows you, because that's blocking a lot of the presentation.
Oh, yeah, is it this one?
Yes.
This is it?
Yes, sir. Thank you.
I can close this, or?
No, please just drag it down, sir.
Okay.
Thank
this
you.
Is better, right?
Yes,
yeah.
Yes. Thank you.
All right. So the immune system, apart from eliminating microbes, it senses tissue injury, it helps in repair, and it also provides a certain amount of normal function which is required for tissue homeostasis. So therefore, studying the immune system and its dysregulation is one of the key components of studying human disease. So, and why do we need to know this immune landscape? So this immune landscape tells us what kind of drugs we can have for what kind of tissue, what kind of disease, and what kind of disease stage. So, for example, current management actually works on immunomodulation, and for most of the ocular diseases, but it's often restricted to the use of steroids, which is essentially very broad. It is not very targeted. So therefore, a lot of us scientists and clinicians have been working on identifying the biological basis of this So to find out biomarkers and therapeutic targets so that we can move towards a targeted therapy. So this is quite the goal of biomedical research the world over. Now, how are we going to tie together all this knowledge for treatment of ocular conditions? So we have the disease pathology, and inflammation is sort of the core of almost all diseases. So we need to know about the interactome of how immune cells, secreted factors, the local tissue, the ocular fluid, and the microbiome, the microbes that reside in the periocular space, how all of these come together to modulate the disease pathology. And this has applications across a variety of different conditions. So the paradigms for human health have been evolving, and the primary reason for that obviously is that 50 years ago the lifestyle was quite different. The microbiome of our bodies, of our tissues, has been quite different, and this has been a function of the diet that we use, of the lifestyle, whether we were more active and now we are less active. Because of COVID, we are even doing webinars sitting at one place, we are not traveling anywhere. So all of these put together has led to a different variety of symptoms. Even if the disease was already there, we have newer symptoms, we have sort of altered responses to the drugs that we had previously, which has forced us to come out with new medications, to talk about new comorbidities, etc. So this is an important driver of the research that we do today. So at our laboratory, we work on a variety of aspects of a disease, and this slide essentially explains how we do all of that. So in ophthalmology, imaging of the eye is a major component of analyzing and diagnosing and monitoring disease. So we start with that, and we collect tear fluid, epithelium, tissues and cells, as well as blood from various patients after they consent to being part of a study. And then we analyze gene expression, we analyze secreted proteins like cytokines, inflammatory factors, growth factors, etc. And then we, from the tissues that we get or from the blood that we get, we do high throughput gene expressions. It can be a microarray, RNA-seq, or even targeted multiplex analyses. Of course, a lot of sequencing in order to look at genes or mutations underlying these diseases. We do some epigenetic studies. We are now doing a lot of immunophenotyping, where we are analyzing the immune cells that are coming and going into the eye and outside the eye. And we'll touch this briefly before we dive into the microbiome part of it, because it's important to understand that while the eye is immune privileged, there is still a lot of activity happening in and around the eye. Then we have proteomics, we use mass spectrometry. We have a lot of these platforms which are in vitro platforms, which analyze different pathological features of disease. So we have cells from the corneal epithelium, retinal epithelium, stroma, trabecular meshwork, etc. So different tissues have different cells, and we grow those cells in different with different stimuli and stresses, and study how they respond and which part of the disease pathology they can mimic. And that's when we have a disease model in vitro that we then try and treat with different drugs. So finally, then we come to the microbiome, and one of the latest things that we are looking at are cellular energetics and metabolomics, how each layer of the eye, their metabolism gets affected as you have in different kinds of disease. And you put all of this information together and integrate it with the clinical parameters, and then you have a highly coherent integrated data set of human tissue analysis and the disease phenotypes to target and find new drug targets. So what sort of samples we collect? We have vitreous humor, aqueous humor, tear samples, swabs. All of these are frozen, collected and frozen, and then we do a variety of omics, right? So we do transcriptomics, we do proteomics, we do immunophenomics, and all of this data or the signatures are analyzed together to build a knowledge base, which then helps us identify druggable targets or molecular diagnostics, which can help us monitor disease. All right, so coming back to the central pathological feature of almost all diseases, which is inflammation. So inflammation is primarily mediated by inflammatory cytokines. So things like TNF-alpha, IL-6, VEGF, these are words that you would have heard definitely. And these lead to immune cell infiltration, right? Macrophages, neutrophils, etc. And activation of tissue-resident immune cells. So these all lead to activation of complements, can lead to changes in vascular permeability, can cause edema in the tissue, leukostasis, and all of that can lead to the disease pathology that you see in the patients. So this is just a fun video to show you what inflammation actually does to the cells. So pay attention to this. So this is a live imaging of cells, and you can see the nuclei, you can see the intracellular organelles. These little dots are mitochondria, Golgi bodies, pieces of ER, etc. So this is under normal conditions, that epithelial cells where there is no stress. Now, to the same cells, when you give TNF-alpha, which is an inflammatory cytokine, what you will notice immediately is that the cell's shape has changed. The cells are hypermotile. They are running all over the place. Whereas here, the cells were quiet and sitting in one place. So you can see what an effect inflammation has. And this is very important. Biologically, if you think about it, you get inflammation when the tissue is hurt in some way or there is an infection or something. You need the cells to come there to repair the area and go back to homeostasis. So that puts an immense stress on the cells. So this, when it goes on unchecked, is when it causes chronic diseases. So who are the responders to inflammation? To the immune cells of the eye. So you can separate them into two main groups: the residents, which are the tissue resident cells. So these are dendritic cells, macrophages, the regulatory T cells, which are called the gamma delta T cells, the mast cells, which are sort of the first responders. So they release things as soon as the tissue gets injured or infected. And then you have the visitors. These are the ones who are surveilling the tissue, making sure everything is okay. And these are the ones who will accumulate in the tissue if the tissue has a problem. And these include neutrophils, type 1 type macrophages, natural killer cells, a variety of T cells, and B cells, which will ultimately produce the antibodies. So if you have a dysregulated proportion, a higher proportion or lower proportion of these cells in your tissue for a long period of time, you're going to get disease. So where do immune cells come from? And this is where the concept of immune privilege of the eye gets challenged, because we and other people have shown that the eye, although immune privileged, in case of disease is no longer as privileged. There are immune cells that do come into the eye, and here this is in green are cells that are neutrophils that are stained with the surface antigen called Ly6G, and this is seen in an animal model. So this is the wild type, and this is a knockout animal model which gets a disease which mimics the human age-related macular degeneration, and you can see that immune cells do come into the retina, and this is a snapshot of a human AMD retina which shows the same kind of cells coming in. Now, we did an experiment here where the immune cells were labeled. Okay, so when you activate the immune cells and you have labeled them with something that gives off a signal like GFP, so here it is RFP, and found out, and we looked at the eye to see if these immune cells which have been put in circulation can actually go reach the eye and get into the retina. So this is a video that will show, and what you have to keep an eye out for are these little white speckles. Okay, so these little white speckles are the fluorescent labeled immune cells, and this is a 3D rendering of the mouse eye. It's called ribbon scanning confocal microscopy. So what you will notice shortly is, so now we are going across the layers of the eye, and keep an eye on these speckles that will show up. So now we are looking at the back of the eye, and you can see these speckles show up. And we will go across the different layers, and you'll see the speckles show up at the blood aqueous barrier. So that the previous one was the blood retinal barrier, and this is the blood aqueous barrier, and you have cells infiltrating from both sides. So which means that under disease conditions, the immune cells can enter the eye through the barriers, through the gates, and this is just, we have marked in green all the immune cells that entered this eye. So this is definitive proof that the breakdown of immune cells of the natural barriers allows the immune cells in, and that's a problem. So now we enter the microbiome world for the eye. So most of the microbiome studies, as you know, across all the different conditions that have been done, refer to the gut microbiome, because that's where everything started from, and that's the proof of the pudding, so to say, that you have poor eating habits, you get all kinds of diseases like heart disease, diabetes, cholesterol, high cholesterol, etc. And that is quite well proven that if you can improve your diet, you can get rid of certain things, you can control all these diseases. So the gut microbiome really helps control the host metabolic profile, which in turn helps control the immune system, and has been shown in many different things, including tumors, neurological diseases, etc. So for the eye, there's a huge emerging field of the link between the gut and the eye, particularly for the retina, where people have already shown that gut microbiota and the change in the gut microbiota actually have links to age-related macular degeneration, diabetic retinopathy, glaucoma, uveitis, and ocular surface conditions. So the challenge, of course, when working with microbiome is how you collect the samples and how you analyze the data that comes out of it. So I'm going to cite some important people whose work has been shaping this field. So this is Dr. Maria Grant, and she has been working on diabetic retinopathy and how the gut microbiota actually regulates our retinal degeneration when you have long-term diabetes. So she's made some seminal discoveries on this, and this again, this is Alan Taylor, and he is again looking at the link between the metabolome of the eye and the gut microbiome, and how the diet can actually lead to a change in the microbiota, which can lead to a different set of metabolites, which can in turn lead to disease regression or disease progression, depending on which way you go. And this data comes from animal studies, where you can control what kind of diet is going in. So across the different diseases, across the different organs that are being now affected by how your microbiota looks, how your microbiome alters, the dysbiosis. In fact, in case of preterm births, so in India, we have one of the largest proportion of preterm births, and there's a disease of the eye which affects a high proportion of preterm births, which is called retinopathy of prematurity, or ROP. And what people have shown is that in ROP, because of the fetal microbiome, which in turn is affected by the mother's microbiome, that shows a distinct association with ROP. So these are the different kinds of bacteria that have been found to be associated with these preterm babies developing ROP. And what was surprising is that this link is a modifiable link. So if you can change the microbiota, then you can probably reduce the risk of retinopathy of prematurity. Now, mind you, ROP is extremely dangerous. The child can lose their vision permanently for life. Very large program across many states and many districts of Karnataka and India, which has been adopted elsewhere also. It's called the Kid Drop program. If anybody is interested, they can look it up. So where preterm babies are screened immediately, and there is a small window of a couple of weeks during which a small laser surgery can fix the disease. But if not, it carries a huge risk of blindness for life. Another disease we talked about is dry eye disease, and that has been shown that the reduced amount of microbiome diversity is actually associated with dry eye disease. So which means that these missing populations of bacteria are actually associated the metabolism in a way which affects the eye, the surface of the eye, probably the contents of your tear film. So all these roles that people have been showing are with the gut microbiome, and how changing the gut microbiome can possibly alter the metabolic and immune profile, which then can actually regulate the disease progression. An interesting study, this is in mice, is how light can regulate microbiome composition. So this is going looking at the problem the other way around. So where the light that's entering the eye, and you have these light sensitive retinal ganglion cells that actually control how we sleep or wake, our circadian rhythm, and these actually can help regulate the composition of your microbiome in the gut. So that's like the full circle, the gut regulating things in the eye, and the eye can in turn regulate things that can change the microbiome in the gut. So quite fascinating. There's quite a few of these studies that are going on right now, where a lot of age-related diseases, I did across cross-sectional samples, across twins, and so on and so forth. Again, this is another study in a syndrome called the Stevens-Johnson syndrome, which has a severe ocular surface condition where patients can go surgical interventions and very, very strict monitoring and treatment. So people have identified specific bacteria, and they've looked at that across these different conditions. So this is SJS Stevens-Johnson syndrome, this is graft versus host disease, and this is dry eye disease. But this is a study where they looked at only a specific set of bacteria to see what kind of differences they can find. Now, it's important to remember that this study is done on the samples from the eye. Okay, so this is not bacteria from the gut. These are samples that have been taken from your eye and have been studied, and wherein lies a big challenge. So the samples from the eye are going to be very tiny, so analyzing them requires a whole different level of complexity, and there are certain levels, fine levels, to which you cannot go because you just don't have enough material microbiota. This is another study on post-fever retinitis. So retinitis is inflammation of the retina, and that can be caused by a variety of different viral infections, and this can often lead to such bad inflammation that your retina can completely degrade or detach, and that can lead to blindness. So a variety of viral, as you can see, a variety of viral viruses have been tested by PCR, and to look to make sure that this is the samples that they took are of retinitis that has been caused by bacteria, and they have isolated and looked at the bacteria that have caused these. So a lot of the microbiota in the eyelid, which is contiguous with your skin, obviously is shared between the skin and the eyelid and the conjunctival sac. So that is something that can confound your studies. So when we are looking at ocular surface specifically, then we have to take that into account. So an important study that was published by Rachel Caspi's group quite recently is this link, and this is where we are now entering into how the microbiome can actually interact with the immune system. So here, what they were able to show in an animal model of autoimmune uveitis is that the microbiome can actually activate an autoreactive T cell receptor, which causes these T cells to migrate into the eye and cause the uveitis there. So the uveitis is inflammation inside the eye, and that is being fed because the T cells are being continuously activated by an altered microbiota in your gut. So now you have a link between or a functional explanation of the link between the eye and the change in microbiota. So the bacteria have the property or can develop the ability to activate or dampen your immune response. And like I pointed out, that's very important in a variety of ocular diseases. So this is more data from that same paper where they found these retina-specific T cells which were activated in the intestine, and these activated T cells, they crossed the barrier into the mouse eye and then eventually caused the uveitis. So this is what we mean when we say that the tolerance or immune privilege has been broken down. So On the ocular surface, on the front of the eye, there's an immune landscape as well. So we study that by taking these ocular wash. We take Schirmer strips and surface swabs, etc., from which we do the microbiome studies. So these, hold on, this is gotten stuck again, I think. There we go. All right, so these are studied using flow cytometers. So this is a Luminex, this is a multiplex assay system, and this is how in flow cytometry, your cells pass through the they are stained by certain kinds of markers, or if it's an antibody, it's there's a bead attached to it which gives a fluorescent signal. All right, so a condition that affects us most often is dry eye. We talked about it. Eyes hurt, feel tired, etc. And you get these typical staining of the eye. If you go to the doctor, he'll stain with fluorescent dye and say that, okay, you have dry eye disease. So this is another group, Kelly Nichols group, and they studied the microbiome of the eye. So here they have actually taken the samples from the eye, done the sequencing, and then found that there is a unique microbiome which is different and distinct between the dry eye and the control. So this is, if you look at D, so these are various statistical analysis to look at the diversity of the data. And what you can see is in this Spearman plot, you can see that the greens and the reds are different. So this gives you an idea about the diversity. What was interesting, what they showed, and this is something that we found in our own data, is that if you rinse the eye every day, it doesn't really change your microbiome. So if you think that washing your face every day is going to change the microbiome, it doesn't happen like that. What it also shows is that the microbiome so this is another set of analysis to show that they are discriminant. So they are distinct between dry eye and control groups. And so I'll not get into too much in depth of it, but here they looked at hold on, I should check the chat, is it? Okay, I'll answer these later then. Okay, so the network analysis, this is done in different ways to look at the enrichment of the different genera, different phyla. So in these kinds of analysis, so they have done the V4, V5 sequencing. It's a similar approach to what we also use. That doesn't give you species level, but it will give you from phyla all the way down to order. So, okay, so now I have introduced you to microbiome at the local tissue. We talked about microbiome in the gut and how it regulates the disease in the eye. Now I've shown you some evidence that there is a microbiome difference when you have at the eye as well, if you measure the microbiome, the metagenome at the eye. And now I'm going to bring you back into how the immune system in the front of the eye and the microbiome can interact. So in order to maintain healthy ocular surface, we have to maintain this dynamic interaction and homeostasis between the corneal cells, the trafficking immune cells. So I'm going to introduce you to another disease called keratoconus. So dry eye, we just got introduced to. Keratoconus, I will introduce you to it. And we have previously looked at how the ocular surface immune cells, so we take sample from the ocular surface and measure what kind of immune cells are there. And what we find that indeed there are distinct differences between these two different kinds of diseases, although they are both diseases of the front of the eye. So keratoconus is a disease which causes thinning of your cornea. So the thinning of the cornea leads to these sorts of symptoms like eye strain, ghosting, especially at night. You see double, triple headaches, general eye pain and irritation, and can also cause eye rubbing. In fact, eye rubbing can cause keratoconus also. So what this leads to is this appearance of the cornea. You can see that this is a normal cornea and this is a keratoconic cornea. So you can see the conical shape and therefore the name keratoconus. So what it causes is that in the initial stages, you'll have this blurriness, you'll have very rapid changes in your glass power, and eventually in the later stages, glasses will not be able to give you proper vision, and you'll have blurry vision all the time unless and until you get a transplant of your cornea. And currently, surgery is the only treatment. So our group works on this very heavily, and what we had found is that inflammatory cytokines like MMP9 and IL-6 across the grades of keratoconus, so they are graded clinically, they keep increasing, right? So as the disease increases, you have more and more inflammation. And what this inflammation causes is it causes a loss of this important enzyme called lysyl oxidase, as well as collagens. This lysyl oxidase actually is required to link the collagen fibrils side by side, which gives you transparency and gives the cornea strength. When the lysyl oxidase is not there, the cornea becomes floppy. It doesn't have strength. So that causes the pathology. So again, there's an underlying chronic inflammation that is going on. So we had already shown that there is a different tear film biochemical status in keratoconus. We have also seen that there's a difference in the immune cells that are coming there. So now we wanted to look at what is happening to the ocular surface microbiome in this case. So we started by taking samples from the same group of patients where we took an ocular swab, we took an ocular surface wash to analyze the immune cells, we took Schirmer strips to analyze the tears, and the swab was then processed to extract DNA, and then the V3, V5 sequencing was done, and the phylogenetic trees were constructed based on that. So essentially, you would have studied that the way the phylogenetic trees are classified is based on sequencing of conserved regions across different species, and that identifies the different phyla, class, order, family, or genus. But for species level, you have to go down to whole genome sequencing, which we found was very difficult to do in case of the ocular surface, because the sample was very little and it was heavily contaminated with, not contaminated, you'll get a ton of human DNA in it. So the problem was that almost 97 to 99% of it was human DNA. So it made identifying species very, very tricky. So in our data, we did this, and what we found were 515 different genera emanating from 21 different phyla at the ocular surface. So these are donut plots, and you can see immediately in case of phyla that these are controls. So these are our healthy controls, and these are keratoconus. And you can see suddenly something has appeared here. Of course, the proportions of the rest are also changing, but this is very stark. When you go down to order, again, you can see that something new has appeared, and there are a variety of different orders that are actually increasing or decreasing with disease. So, at the same time, we analyzed the immune cells from the exact same patients, and what we found was that activated neutrophils, these went up with the disease. So did T cells. Now, if you remember, there was the animal experiment showed that in the gut, if you modulate the microbiota, it can activate T cells, and that can go and cause uveitis or inflammation inside the eye. So we are starting to see some parallels of it in actual human patients here. So then we looked at inflammatory factors. So again, like your MMP9 is very high. You have a bunch of different inflammatory factors, IL-6, etc. These are very high, and obviously these are what's calling in all these immune cells as well. So when you put all of these things together, what you find is a distinct set of organisms which are going down in disease. So potentially these could be protective, and a distinct set that goes up in disease. Potentially these are the ones which can then modulate these inactive neutrophils into activated neutrophils, or quiescent T cells into activated T cells, and cause the tissue to start producing more and more of cytokines, etc., which will call more of these immune cells there and set off a vicious cycle that will lead to the disease. So the emerging paradigm now is that the corneal and the conjunctival epithelium cells, as well as the inside the stroma, we have nerves, etc., and that's a different story. I'm not going to talk about that. But these three we had shown to interact, to actually show the dysregulation of the immune phenotype in these diseases. Now we are adding on microbiome to it and are showing that there is a distinct set of microbial species that can affect this immune cell and therefore affect the ocular health. But what's the clinical implication of this? So again, it's a donut, and you can see that there are certain things that are available, that are showing up in this analysis, and certain things that don't show up in this analysis. Some of the arrows have moved. I apologize for that. But let's focus on one thing here. So this is the order level analysis. And we are going to look at this particular genus, which is Clostridia. So Clostridia are already associated with a variety of different chronic inflammatory conditions. They have been shown at the gut level, they have been shown to be associated with cirrhosis, diabetes, autoimmune liver diseases, and mucosal microbiome. So what we are are now showing is that this may be stimulating a specific subset of T cells, and this we found by statistical correlation analyses between the immune cells subsets and the different species, different genera that we found in our sequencing. So this could be a possible target, because what we know from our previous work is that this MMP9 that I showed you that goes up, the lysyl oxidase that goes down, MMP9 is actually activated by this particular cytokine called IL-17A, which we showed also goes up in keratoconus. And more recently, we're just going to communicate this paper where we show that this transcription factor, ROR gamma, actually transcribes IL-17, which then activates MMP9, which then represses lysyl oxidase, which then causes keratoconus. What's interesting is that Clostridia can regulate ROR gamma and ROR gamma T. ROR gamma T belongs specifically to the ROR gamma that's in T cells. So therefore, this gives us an opportunity to have a possible therapeutic intervention, because Clostridia, there are already specific antibiotics that can work on Clostridia. So while there are other drugs and other therapeutics that we are working on, like inhibition of MMP9, activation of lysyl oxidase, etc., this provides us with a completely new paradigm for treating ocular surface disease by targeting the microbiome. So therefore, our evolving paradigm for clinical pathological mechanisms is that we know that we call it the keratoconus inflammaxis, where you have NF-kappa B activation, which leads to MMP9 and IL-6 secretion, as well as influx of reactive oxygen species that causes the damage to the ocular surface, and that reduces your lysyl oxidase and collagens, that further weakens the cornea. So there is a biomechanical weakening of the cornea, and that is what is causing this disease. Now we are adding to it that this secretion of MMP9 and IL-6 is probably dependent on a cyclical activation, a chronic inflammation caused by immune cell subtypes, which seem to be regulated by altered microbiome at the ocular surface. So what I've told in this last section is this novel paradigm of immune regulation at the surface of the eye directly by the local microbiome. So this will have great implications in disease treatment in the future. So to end my talk, I'll just talk a little bit about SARS, because nowadays everything is about SARS, so we work on that quite a bit as well. So there has been evidence in the literature recently that shows that there is you can detect the virus in the tears. You can there is a possibility that the virus can be transmitted from eye onto the nasolacrimal duct down to the nasal mucosa as well. And remember that the ocular surface is a mucosa. So what we did was we categorized the expression of the ACE2 receptor and TIMPRESS2, which are the key modulators, or rather the host factors that are required for virus entry and virus uptake and processing. So what we found was that across these different layers of the eye, so starting from the retinal pigment epithelium, retina, trabecular meshwork, we find that the corneal cells, particularly if you look at the corneal epithelium, the corneal, so this is your corneal epithelium, this is your conjunctival epithelium, these have the highest amounts of expression of your ACE2 receptors as well as TIMPRESS2. Now this is important to note that the RPE and the retina actually has several log4 lower expression of this. That's because the corneal and conjunctival epithelium are your ocular surface mucosa, and all the mucosal surfaces express these ACE2 receptors. So this has been shown by another group as well, where you can see the ACE2 staining in the cornea and the limbus. This is in cadaveric tissue, and we also have shown the same. Our publication is in communication right now. So this shows that the ocular surface can serve as an entry point for the virus. So we all have to take care about it. And therefore, from our group, we have been publishing quite a bit of information on how to look at workflow in the eye disease hospitals. So how to handle conjunctivitis, etc. during this epidemic. So if anybody wants any information regarding that, they can look up these papers. So with that, I'll end my talk. I'll thank my team, my clinical team, my funding agencies and collaborators, and that's our department. So thank you. I can take your questions now.
Thank you so much for that informative talk, sir. It was enlightening to learn about the ocular world, which most of us take for granted. I'm sure that it has sparked curiosity in many of us here, and there are a lot of questions waiting to be asked. So let us begin with the Q&A session without any delay. The first question is from Dr. Beatrice Maria Sequeira. If there is a link, will not the T cell circulate back to bring about killing and inflammation too in the gut?
Yeah, certainly. So the circulation is definitely there, which is what we were showing in that video also. So if you have things circulating in the body, that does circulate into your eye, onto your ocular surface, to the back of the eye, etc. So we do have these uveoscleral lymphatic channels through which these immune cells can go, and then these immune cells will come into your draining lymph nodes. So that connection is definitely there. Now, the T cells that we are talking about here, so the cytotoxic T cells, the macrophages, etc., which are part of your immediate response, are the good ones. So they, as soon as you get a big infection, they'll come in, they'll eat up the bacteria, and then help you go back to normal. But what we are looking at here is beyond the infection kind of situation. What we are looking at is dysbiosis, where your normal flora has actually changed, and that is causing a low-grade chronic inflammatory activation of your T cells, and now these T cells are going into your eye and causing destruction to your tissues rather than going and eating up bacteria. Because at these places, there is a certain level of microbiota. So typically, an infection means that if it's from an opportunistic pathogen, then it's coming from out. But if it's one of your own pathogens, that is because your immune response is lower, suddenly grows out of control. So that is a different situation. Here we are talking about where the overall complexity, the composition of the local microbiome has actually changed. So therefore, maybe the T cells are still or the macrophages are still killing some bacteria, and maybe they are killing the wrong kind of bacteria, the ones that protect you. But that's one aspect of it. The other main aspect of it is that the microbiota is somehow activating these immune cells and making them go into the eye and cause destruction in the eye.
Moving on to the next question. Tears contain lysozyme. Would continual flow of tears affect the microbiome of the eye? By Arun Kumari.
Yes. So apparently it doesn't. So that's the observation that we've been having. So we thought initially that because of the tear film production and our activities through the day, the overall microbiome would actually change from day to day if you were to look at it, but it doesn't actually change. That's what was quite interesting to see. In fact, things that was in the dry eye paper, where they actually looked at people with or without eye washing, and they found that the microbiome didn't really change even with eye washing.
The next question is from Madiha Khan. Do genetics also influence our microbiota?
Oh yeah, I would say so. So the microbiota also depend on, and the reason they colonize wherever they are colonizing is because your tissues are also providing them some food, and that comes in terms of metabolites typically that they use to grow. So if you have some genetic mutations in any of these metabolite pathways, which a lot of us carry, not all of us express the same proteins in the same levels. So that would definitely change the levels of those metabolites that these particular bacteria need in order to grow. So they may be overgrowing or undergrowing, and that might lead to the change. So definitely genetics would have an effect on the microbiota.
The next question is from Dr. Beatrice. What is the percentage of similarity of the microorganisms in the gut and in the eye?
So that is a study that we haven't done yet. Ideally, you need to do that from samples taken at the same time from the same patient. Cross-sectionally, if you do it, yes, there's quite a lot of similarity. This is just subjective analysis because we haven't done that concurrently collected sample thing. But what we can see from the data that's been published is that with the skin, there's quite a bit of similarity. With the gut also, but the proportions are quite different. So you do have the similar kind of bacteria, but the proportions are widely different.
Moving on to the next question by Shagla Sharma. Well, one second. In general, what efforts can be made to overcome dry eyes disorder, or simply is there any possibility of bringing microbiota back to normal?
Yeah, so that's what we are all trying to do now. So these things need to go into a proper randomized clinical trial now. That's what we are also planning on. The severe dry eye disease is a major problem actually. Dry eye is present in about 50% of the population nowadays, but the severe dry eye, which is about 10 to 12% of that population, they really can't see, and it's very difficult to treat also. So we have come up with a variety of different treatment possibilities. There's one eye drop that recently came out of work from our lab. It's a simple disaccharide trehalose, which seems to work well with subacute dry eye disease. In acute dry eye disease, you really have to treat them with steroids and things like that. Now that we know a little bit more about the kind of immune cells that are coming in, we can have more targeted therapies. So for dry eye, currently in the market, you have steroids, you have Restasis, which is cyclosporin. You have a new drug that has come, but that needs proper diagnostic testing. That's ICAM LFA blocker. And then there's at least half a dozen different drugs across different cytokines, IL-1, then IL-6 inhibitors, IL-1 inhibitors, etc. So antibody-based as well as small molecule drugs, which are being tried out. Apart from that, there's actually quite a few interesting therapies that are coming, which is light modulation. We were talking about how the light can change the gut microbiota, and I wonder if that can be the case with the ocular surface microbiota also. So there's a treatment where they'll put a light mask on you, where there's a certain a light of a certain power and wavelength that helps increase your tear production and stabilize your tear film so that your dry eye symptoms go away. So so maybe light-induced microbiome changes that can help dry eye disease. I don't know. We'll have to study those. And the antibiotic part of it, that field is still emerging. So we need a lot of more studies like this. The studies that I showed you are the only studies that are there right now. So that's why it's we need more data to come to consensus on which exact groups of microbiota we need to target using which kind of antibiotics for these specific diseases.
The next question is from Dr. Sai. What impact does laser treatment have on the microbiome of the eye?
I would like to know. I haven't done that study yet. Yeah, the ocular surface microbiome, like I'm telling you, there's only one published paper and ours will be the next one. So we haven't done it yet, but I'm sure lots of people are now doing those kinds of things, because Laser treatment also sometimes causes these problems like dry eye disease. Sometimes it can be severe. That's like 1 in a million kind of chance where you can get worse vision after the surgery. That's caused by ectasia or subepithelial scarring. So those can also be linked to microbiota, I'm sure. But those kinds of studies need to be done. Not sure, but yeah, I would say that because the laser would cause a change in the inflammation and the inflammatory status at the locality in the cornea. So I would say the microbiome should respond to it. So if you don't have a healthy microbiome, maybe it'll exacerbate and give you dry eyes. But if you have a healthy microbiome, then maybe it'll resolve and you'll have a happy time seeing without glasses.
Can the way, Ritika Keshav asks, can the way of removing your contact lenses affect the microbiome of the eye? Sorry, it causes keratoconus. She's asking, can the way of removing your contact lenses have an effect on the cornea and thereby
that is not associated with keratoconus. And in fact, rigid contact lenses are a form of treatment for keratoconus.
Moving on to the next question by Dr. Syed Wajid. Can microbiome analysis of the eyes tell us their health status?
Yes, I would presume so, but more data needs to be accumulated. I think more number of studies as they come, we will know about it. But definitely right now we are seeing the association, right? So I would say that there would be eventually a definition of what should be the normal proportions of which kinds of microbes on the ocular surface. That would be, and any deviation from that would cause disease. So that information needs to still come out. Research is still going on.
Nishi Mehta asks, in your opinion, can information on the eye-gut axis make it possible to use personalized medicine to treat ocular disorders?
Yes, that's what I was talking about at the end of it, right? So I picked up one specific genera for which there are already quite a bit of antibiotics available. So the idea would be to adapt some of those into an eye drop and try and see if that can help treat keratoconus. So I would assume that would be doable for a number of other diseases.
Ramya Ji has a question. Was there any change observed in other ECM proteins and also cell anchoring proteins?
Yes, so a lot of proteins change. We have done proteomics, so a lot of pathways change, a lot of proteins change. The ones that I showed you are the ones, just to keep things simple. So essentially, a lot of ECM related proteins directly and indirectly are changed in the disease, because this disease is primarily a problem of the extracellular matrix structure and integrity. So what happens in the disease is that the collagen lamella, because they are not properly cross-linked, they slip. And then because inside the eye, in order to maintain the shape of the eye, because without that you won't be able to focus the light, so you always have to maintain the shape. So there's a lot of pressure inside the eye. So that pressure, if the collagen is not properly bound across its fibrils, that structure, that strength is not there, it will bulge out, and that's what's driving the disease. So apart from the ones that I've mentioned, there are quite a bit of more things that are altered, glycosaminoglycans, for example, TGF-beta related proteins, for example, they are also altered.
Namrita Zakane asks, is chronic blepharitis a consequence of dry eyes? How can we treat it?
Blepharite, I think it's the other way around. It's very difficult to tell which one comes first. But yeah, blepharitis is on the surface of your lid, but that also causes problems inside the lid, and the problems inside the lid cause dry eye. Sometimes dry eye can cause the problems inside the lid, and that causes the blepharitis. So the blepharitis treatment typically involves cleaning your eyelid. They'll give you some shampoo or something, making sure that you don't have dry scalp, and keep your hair and everything very clean. So that's the treatment for it. And if it is very bad, then they'll give some topical steroids or immunosuppressants like tacrolimus or something like that.
Ms. Shalmali Kamath has a question. Does the radiation coming from screens affect the normal microbiota of the eye?
I would assume so, because over time, I'm pretty sure that does affect the dynamic balance between the secreted or soluble factors in the eye, in the tear fluid, etc., and the microbiome that's there in your conjunctival space and on your surface. So the association of dry eye disease and computer vision syndrome is quite well set. In Bangalore in particular, a lot of the people who come with irritation of your eyes and feeling of dryness and grittiness like sand in your eye or ocular pain, they're mostly associated with overuse of phones and laptops. And not taking breaks and lubricating your eye enough. So definitely that dryness, that change is going to cause the change in the microbiota. Now, whether or not the blue light and the wavelength of blue light that's emanating from the screen is directly affecting the growth of certain kinds of bacteria, that I think is an interesting study that needs to be done.
Moving on to the last question by Namrita Zakane. Carboxymethyl cellulose is prescribed mostly for dry eyes. Is it efficient?
Yeah, so CMC is essentially to sort of bring back the thickness of your tear film. So in dry eye, what happens is that your tear film has three components. So you have a mucin layer at the bottom, then the most of it is filled with an aqueous layer, and then there's a lipid layer. Right, so it's a tri-layer, so that the tear film that is produced, it always maintains a certain thickness covering all parts of your eye. The moment you remove that, your underlying epithelial cells, they start getting affected. They start dying off because they get inflamed, and that's what causes the dry eye. So what CMC does is it sort of creates this layer again, but it doesn't actively block any inflammation or anything. So giving CMC to an aqueous deficient dry eye patient is not going to fix it. But if you just have mild itchiness and a little bit of blurriness or pain or whatever, people always try these frontline mild things like tear substitute, CMC, HPMC, etc. Sometimes they work. If they don't work, then you have to get on to something a little stronger.
There is one more question. Does the use of contact lenses negatively impact the eye microbiota of an individual?
I would say absolutely, yeah. I would say that, well, whether or not it's negatively, I don't know. Again, like I pointed out, the local microbiome studies, we are only the second group in the world doing it. So, but there are other groups, I know at least 3 other groups in China who are also doing different things. So all those will come out. So we will know. But right now I don't have an answer for you.
Thank you so much for answering those questions patiently, sir. With this, we come to an end of the Q&A session.
Great.
Thank you, participants, for the well-thought questions. I'm certain that it has helped many of us to learn more from Sir's talk. Participants, I'm sure that all of you enjoyed this session. We will start our next session at 2:45. Please stay logged in, for we will begin shortly. Thank you.
Thank you.
Thank you, Dr. Ghosh.
Thank you. Goodbye.
Thank you, Dr. Ghosh.
Thanks.
ಹಲೋ ಏನಿಲ್ಲ ನನ್ನೊಂದು ಲೆಕ್ಚರ್ ಇತ್ತು ಕನೆಕ್ಟ್ ಆಗ್ತಾ ಇಲ್ಲಲ್ಲ ಈಗ ಆಗಿದೆ. ಲೆಕ್ಚರ್ ಕೊಡಬೇಕು ಸೇಂಟ್ ಜೋಸೆಫ್ ಕಾಲೇಜಲ್ಲಿ ಆಗಿದೆ ಈಗ. ಓಕೆ ಸರಿ ನಾನು ಇಲ್ಲೇ 3rd ಫ್ಲೋರಲ್ಲಿದ್ದೀನಿ. ಓಕೆ ಫೈನ್.
Good afternoon, Dr. Nitin.
Hello, good afternoon.
So internet is working well now?
Yes, it did work a bit. Now, how do I share my slides?
Sir, so there'll be a student moderator. They will give a brief introduction about you, after which I will give you presentation rights and you can share your screen, sir.
So once you give me share rights, then what do? I go to my thing and open that thing?
Yes, sir. You on the bottom of your screen, one option will come in between participant and chat. That will show share. So from there you can select, you can press share and then select desktop and share your screen, sir.
Oh, okay, fine.
Yes, sir.
Dr. Nitin, shall we start the session?
Yeah, just 1 minute.
Sure.
Let me open my this thing. Desktop. Yes. I'm okay.
Calvida? Yes, Calvida, you can just start the session, ma'am.
Ma'am, it will be Sumeru who is continuing.
Yes.
If you can't fly, then run. If you can't run, then walk. If you can't walk, then crawl. But by all means, keep moving, said Martin Luther King Jr. This is indeed a wonderful occasion to remember these words as they were spoken to an audience of students, just like today. Good afternoon again to one and all present here today. I hope you enjoyed the previous session. We have with us today another incredible speaker who will most definitely engross you with his vast knowledge of the gut. I have the honor of introducing to you the highly accomplished Dr. A.R. Nitin Rao, Chief of Surgical Gastroenterology at MS Ramaya Hospital, while also Professor of Surgical Gastroenterology at MS Ramaya College, both right here in Bengaluru. Dr. Rao started his illustrious academic journey by doing his MBBS at the University College of Medical Sciences in Delhi. He did Master of Surgery from KMC in Mangalore. He was awarded a fellowship by the Austrian College of Biomedical Sciences in Sydney and was later followed by a membership to the prestigious Australian Institute of Medical Scientists. He later earned fellowships to the Association of Surgeons of India and more recently to the prestigious American College of Surgeons. He is a member of 9 August associations, including International Hepatopancreatobiliary Association, Asia Pacific Hepatopancreatobiliary Association, and the Society for Surgery of the Alimentary Tract, USA. When he was 18, he was awarded National Talent Scholar of Basic Sciences by NCERT and UGC. Since then, he has received a travel scholarship to attend the Asian Society of Transplantation in Malaysia and a travel grant to attend the APWD in Hong Kong. Moreover, he has also written a chapter of the book, Management of Liver Failure, out in English and Chinese. If this isn't grand enough, Dr. Rao has also been actively involved in research regarding the liver, its transplantation, and cancer, having co-authored 42 papers so far. He has been almost to 60 conferences showing his keen interest in the most recent innovations in his field. He is currently working on developing new technologies for surgery and has already developed prototypes. Thank you for being with us here today, sir. Now, Dr. Rao will deliver his talk, Gut Microbiome Changes in Diseases and Interventions, about a topic which would be the central discussion of the scientific community, if not for the unfortunate occurrence of the pandemic. I request all the attendees to type out their questions in the chat box as the session progresses. There will be a Q&A session at the end of Doctor's talk when he will be answering these for you. Without any further delay, let us begin the talk. Over to you, Doctor.
Thank you, Sumeru. Let me try and share my desktop.
Sir, you see that window to your left, right, sir? You can close it.
Yeah, this one.
Yes, sir, you can close that.
Okay. Okay.
Yes, sir, your slide is visible.
Thank you, Dr. Sumeru, and thank you, Dr. Jyoti, for this invitation. I have a disclaimer or disclosure to make. I'm not a microbiologist, so I may miss out on names of multiple of these bacteria so as such, but it is out of keen interest that I look into this area as well. Then I have tried to a review of what is available literature on the net for this presentation. So I have picked up a lot of stuff, what is available on the net or on different papers, etc. To start off, I think we'll just get a little bit basic about what is microbiome, what is omics. So I think Professor Tatu has given a title of omics in this COVID. So we'll differentiate what is microbiome, microbiota, pathobiome. So microbiome is a collection of what all is available in a given area. So when we say gut microbiome, it is related to gut. Oral microbiome related to oral. I mean, any environmental place connected with environment, you can have multiple amount of bacteria, fungi, virus, etc. Microbiota are the actual, what are those particular bacilli or bacteria or virome exactly as that. Pathobiome generally refers to the pathogenic part of these, right? And most of these environments are in homeostasis with the place in which they are located as such, right? So it's not supposed to be harmful for that individual, right? Or organism or organ, etc. Right? However, when this balance sort of mismatches, that is when you say there is dysbiosis. Now, all these living organisms are going to produce lots of chemicals and metabolites. So a study of those is what is metabolome. And if you look at a genetic analysis of the whole contents of this, so we practically, let's say you pick up all the genes that are available in a given sample, so that is known as metagenomics. And if you look at all the proteins, so that is known as metaproteomics. So then there is something known as metatranscriptomics, that is all the transcription from the RNA, etc. that happens, and then you study. And there is something new, cultureomics. I mean, these are all I am learning myself as such, right? So this is a study of all the culture possibilities. What are the different things that you could, media, etc., grow bacteria? And this is a new term I picked up, pharmacomicrobiomes, microbiomics. So that is how microbes in a given environment affect pharmacology of the drug. That is digestion of the drug, absorption of the drug, and processing of the drug, detoxification of the drug, etc. Now, two things to remember before we go on, right? Endotoxins from gram-negative bacteria surface layer generally produce lipopolysaccharides, and endotoxin from gram-positive bacteria, lipoteichoic acid as such. So I looked at gut microbiome, all the publications for the last, from 2000 you will see to 2020, and this I looked up day before yesterday, right? So there are about 32,447 results. But the most important part that you are going to see is the maximum explosion is in the last 4 to 5 years, right? So presently this figure is about 7,000 odd publications in a year. So this gives you an importance of what is the importance of this area and how much people are looking at it, right? So generally the more the publications in a given area means more the interest of the public or the general people in that area, right? So this obviously tells us that, right? So I'll just tell you why gut microbiome importance. On the left, right? So there was a lady in UK who was driving down the car and she was stopped down, and then the police officer did a breath analyzer test and says you have high alcohol levels, so you need to be fined and etc., etc., etc. So the lady said, I've never had a drink in my life. So he said, how can that be possible? It is impossible, right? So obviously she was fined, and then she went back to her physician and everybody and said, I mean, how can I have alcohol in my breath when I don't drink a drop of alcohol as such? So then what worked out, they looked at the gut microbiome in her, especially in her stomach and the proximal part of the gut and such. And something interesting they picked up, that she had a higher level of Klebsiella pneumoniae in her gut, and those particular strains of Klebsiella pneumoniae were producing compounds, especially acetone or alcohol-based compounds, which the breath analyzer picked it up as alcohol, right? So this is one aspect of it, right? So, I mean, importance of knowing your gut microbiome. Of course, it is very rare. And when I looked up at the net, there are 2 other cases reported in Hyderabad where individuals, they have never taken any alcohol, but their breath analyzers were high, but obviously they were not checked for all this. Now on the right, you will see something interesting, that this is, I mean, I don't know those who your foodies, if you have ever read, and you look at the best ever food review show as such, he going having goat poop soup, right? So is he mad that he has goat poop soup, or anybody is mad that they have poop soup, right? But if you look at history, right, long back, supposedly in our ancient texts also, there is a mention of use of cow dung for certain treatments. But in a more formal way, in 1400 odd, there's a Chinese physician who actually prescribed poop of healthy donors to people who are sick with diarrhea and having relief, right? And believe it or not, this is true today, and this is what is done as a medical treatment as well, right? I'll tell you about that much later. Now looking at the gut microbiome, I mean at the phyla level, you have maximum number of them are formicutes, and then you have the bacteroidetes, acinobacteria, fusobacteria, and proteobacteria. And some of them, genus, you important to remember, Clostridium out there, which is important for us. Ruminococcus is important. Faeculobacterium is important. Lactobacillus is important. Enterococcus important for us, right? Allobaculum is important. Prevotella is important. Bacteroides important for us. Bifidobacterium is again quite important for us, and fusobacteria, right? So this is in terms of what is the gut bacteria in terms of numbers as well, right? So in a healthy individual, you are going to find more number of formicutes and then the bacteroidetes in number in our gut. Now, of course, there is a little bit important that you should realize that when anybody does a gut study as such, we generally pick up the feces as a sample, but it has more of the luminal contents. Mucosal surfaces may not present themselves in that amount of quantity as such, right? I think you are all aware of this. I mean, looking at sizes of bacteria, bacteriophages, viruses, etc. But then you look at the numbers, right, in the human body. So about 10 to the power 13 are human cells, whereas 10 to the power 14 are microbial cells. And when you look at it, 10 to the power 12 bacteriophages per gram of fecal material, and 10 to the power 11 bacteria per gram of fecal material. That is what is present there. So how much do we know of our human microbiome, right? We know about 25 phyla are there. There are about 2,000 genera, 5,000 species. About 80% of that is metagenome mappability, and about 316 million genes, right? However, there is a lot of unknown as well, right? There are hidden taxa and strain level diversity, and there are about 40% genes which we cannot match on a functional database, right? Now, when you go through the gut, right? So one is you have the oral microbiota that is in our oral cavity as such, right? But then when you come to the stomach and the proximal part of the duodenum, right, there is very low number of microorganisms. Probably it is related to the high acid content in our stomach. As you go down to the small intestine, that is the jejunum and ileum, they progressively increases to about from 10 to the power 4 to 10 to the power 7 cells. Whereas when you come to our large intestine, where it is heavily populated by anaerobes and large number of microbiomes as such, 10 to the power 12 cells per gram, right? And now this changes with age also, right? So one is the location. So what we believe or expect is at prior to birth, it is a sterile environment, right? The gut as such. And then progressively, for example, you will see in the previous part, there are proteobacteria anaerobes. But then in infancy, bacteroids and bifidobacteria come into place. And then generally by about 3 years of time, we get what is our closer to our adult microbiome sort of picture in our gut as such, right? So then we have more of firmicutes, bacteroids, and by adulthood we have more of bacteroids and firmicutes, right? And in old age it again changes, becomes more obligate and facultative anaerobes. And this is mattered by a lot of differences. Now something interesting for you people to note is, for example, when an elephant calf is there, young elephant calf, right? It needs good amount of bacteria and enzymes for digestion of the huge amount of cellulose and waste contents that it ingests or eats, right? So what it does is basically, I mean, this is known as such, right? The elephant calf eats its own mother's shit, right, to acquire these bacteria. And there are a few other animals as well which do this, right? And this, our gut microbiome changes over a period with many different things that we are exposed to the environment, right? So for example, our birthing process also differentiates what is going to be our gut microbiome consistency as such, right? So whether you are born by a normal delivery through the vaginal route as such, or you are born through a cesarean section, right? Because then the route is different and the bacteria and things that the infant or the neonate faces is going to be different as such, right? Then of course, in the early part of the infancy, whether normal breastfed, child is breastfed or bottle fed as such. Then as we grow up, a lot of other things add on, stress, that is exercise, metabolic, physiological, etc. And very important is diet. Also is a very important part of how our gut microbiome gets constituted, right? Pharmaceutical drugs we take again makes an implication, right? And then geography, because based on our geography, based on our culture, based on our location, right, we eat many different things, different lifestyles, etc., etc., right? So that also is going to define our microbiome as such. And then lifestyle changes. So what is generally a poor quality food is red meat and processed meat, right? Large quantities of that, right? Consumption of high amounts of saturated fats, refined grains, right? So instead of using atta or wheat, you use more of maida, right? So not a good idea. Rice you use, right? Whole grain rice, right? Dehusked, polished, well amount of rice you use, again matters. Sugars, right? High content of sugars in our diet, salt, alcohol, corn derived fructose, right? These all make a change. So if you have more of fruits, vegetables, fibers, white meats, etc., yeah, and then plant derived protein acids, MUFA and PUFAs, etc., right? These are all going to be better for your gut as such. Now this is a very nice thing between a native diet and a urbanized diet. Of course it doesn't matter because so something interesting that some people have studied is very diverse. So most important again is when you look at the microbiome, how diverse is your microbiome. So the more the diversity of your microbiome in your gut, the better it is, right, in terms of health. So people, especially more native, more tribal, generally anywhere in the world, they have wide diversity in their microbiome. But urban based people who have very limited amount of diet, their microbiome changes, right? So for example, let's say a native diet, right? So they will have a lot of phytochemicals, omega-3 fatty acid, they have a high dietary fiber, max and prebiotics, and then they have a lot of short chain fatty acids in their production as such. So what happens? There is enhanced mucus secretion and increased antimicrobial peptides, right? So your gut, the gut barrier that is there between the microbe and other contents and the gut per se is very nice. So there are tight junctions between cells as well, right? So this leads to regulated immune response. So there is decreased in oxygen, and as I told you, upregulated tight junction. So there is a functional intestinal barrier, right? So you are healthy, you have immune homeostasis, right? And then your dietary fiber degrades gut microbiota as well, which are not good for you. But on the other hand, you take a very urbanized diet, right? High in sugars, high in fats, emulsifiers, high red meat content as such, low dietary fiber. There are toxic metabolites, toxic bile acids, leading to hyperglycemia as such. So there is enhanced mucus degradation within the gut, right? And there is reduced antimicrobial peptides. So here, on the contrary, the oxygen levels goes up and there is down-regulated tight junctions. So this is what we mean by saying a leaking gut. So what does a leaking gut means? If your mucosal barrier of the gut is gone, the junctions between the cells are very weak. So it's like having bricks apart. Things can enter through into and then into our bloodstream and into go into our system. So it's a leaking gut, so to say. So there is, and all this leads to this dysfunctional intestinal barrier, right? And then here you have gut microbiota, mucosal degradation, right? So this is a very nice sort of picture which tells you between the difference between homeostasis, that is a well-maintained, healthy sort of situation, and dysbiosis. Dysbiosis means, as we told, the bacterial content, structure, number, etc. has all got varied as such, right? So now when we come and look at this, right? So dysbiosis, as we said, could be because of antibiotic exposure, high fat diet, alcohol, lifestyle, hygiene, etc. And then good amount dietary fiber, resistant starch, probiotics, etc. Right? So as you look at the top, you have a higher number of firmicutes in there, right? And then bacteroidetes, actinobacteria, proteobacteria, etc. They keep going down, right? And then these give us resistance to pathogens. There is immune maturation, good amount of vitamins, amino acids, short chain fatty acid synthesis is going on, and as well metabolic functions are right. But when dysbiosis happens, I mean, you will see here, firmicutes number sort of goes down, bacteroidetes number sort of goes up, actinobacteria is slightly going up, proteobacteria numbers are going up. And this is very well shown in these different, disease condition, right? So for example, on the first left, you will have, for example, obesity, right? You have firmicutes are much higher in number when you look at study the gut microbiome as compared to bacteroidetes. And in undernourished people, you have proteobacteria in huge numbers, right? And believe me, studies of gut microbiome and their changes has been associated with vast number of diseases, even for example going even to the extent of being associated with autism as such. So I think this is the basic crux of how gut microbiome acts and how things sort of change, right? So generally the bacteria, metabolites, they are in a homeostatic situation, right? So in a healthy situation, you have more amount of mucus production, you have healthy gut microbiomes. Now another thing for you people, another thing to remember is when we say gut microbiome, it includes all virus, fungus, etc., etc. But when we look at the studies or what people have studied, generally it is more to an extent of studies related to the bacteria as such, rather than number of viruses or the type of virus, virome, etc. Right? So in a healthy situation, you have more of mucus and then good amount of propionate and butyrate, right? And certain antimicrobial signals leading to good lymphocytes, right? And then gut peptides. So what it does is it decreases the energy intake and decreases blood glucose, right? So whereas on the contrary in metabolic disorders, right? So because these are less propionate, butyrate, antimicrobial signals, right? Gut not there. So there is increased inflammation, there is increased energy intake and increased blood glucose, which is again leading to metabolic disorders. So this is something very interesting, right? So now, as I said, obesity and related to gut microbiome as such. So it has been shown that, so one is the gut microbiome changes. Now the type of the microbiota that are available. So what they do is, if you take two different individuals, right? They may eat the same amount of food, they drink everything, whatever is equal, right? They are given an equal sort of diet. But those who are tending to be obese might be related, especially related to the gut. What it does is the gut bacteria as such, what it does is it absorbs more of calories from whatever food is given. So in an individual who has bacteria related to obesity, if 100 calories are given as a diet, right, he or she may pick up, let's say, about 60 calories. Whereas in the lean person, that is a thin individual, right, same 100 calories is given, he or she may only pick up 10 to 20 calories, right? So, I mean, generally we all, we all limit ourselves to saying it is only the diet, how much you eat, what you eat, right, defines whether you're obese or lean, right? But this is a new thing that has come up, right? That you may have. I mean, it's the bacteria again which decides that what kind of gut or adiposity, etc., that you develop. So obviously these are very complex things, but I will just keep it simple that, the gluconeogenesis increases, lipogenesis increases as such, right? And there is glucose and lipid absorption increase in the body. And there is something known as the gut-brain axis, right? That again decides of what amount you eat and what you eat, right? Right? So that again leads to obesity. Okay, so there has been again another thing that a lot of cancers have been associated, right? So one of the cancers that is well associated with the gut microbiome is the H. pylori, right? H. pylori is in the stomach and the duodenum, and it has been associated with chronic gastric ulcers and CS stomach acid. So that is well known of. But other microbiomes, gut microbiomes, so to say, have not been clearly defined. But yes, if you see, they lead to genomic instability. There is a state of chronic inflammation, immune dysregulation, etc. Right? And these are all dysbiosis states which are likely to lead to cancer-like situations. And colon cancer has been very nicely associated with gut microbiome, especially in the diet that we eat. So consumption of high quantities of red meat, right, and low fiber diet has been very clearly associated with high levels of colon cancer as such, right? So, but there is a multiple factors in that, right? So once the gut dysbiosis happens, that is, as I told you, the number of ratios of these different bacteria changes in number, then they lead on to chronic inflammation that happens and then leads to adenoma. Adenoma is a benign sort of tumor, and then it can lead on to cancer as such. Looking at pancreatic disease, right? Acute pancreatitis, right? So there is a mucosal barrier is gone, and then reduced pancreatic antimicrobial peptides, and there are circulating gut microbiota which are leading to possibility to infections. Now please remember that these are all associations, right? So very few of them have been confirmed. Like for example, as you will tell, no, in microbiology, it has to follow the Koch's postulates, right? So I want to know this, I want to see whether it causes, then it is this. But when we talk of gut microbiome, it's a very complex system because the number of variables, number of things, parameters that get involved are huge in number. So it's very difficult to prove very clearly or even to disprove as such, right? So presently we are in the situation that whenever we talk of these diseases and association with gut microbiome, right, it is related to the number of association. So we always say, yes, this is associated with this disease, right? Of course, gut microbiome has, because there are metabolic alterations and things in the gut, right? And associated with, sometimes in the gut, what happens is something known as a small intestinal bacterial overgrowth. So it is called SIBO. So what happens is, generally, as we told, right, the content of the gut is less in the small intestine. But once the bacterial overgrowth happens, right, so then you needs to burping, flatulence, difficulty, your digestive ability decreases, satiety sort of increase, things like that change. So these are all very typical of what happens in a chronic pancreatitis as such. Now another thing is, gut microbiota can migrate into the pancreatic substance itself and cause inflammation. Cardiovascular disease. I mean, you can imagine that, we are associating gut microbiota with cardiovascular disease. And something which is very clearly associated is atherosclerosis, right? So where there is increased cholesterol accumulation in the arterial wall, right? So there is endothelial cell damage that occurs, foam cell formation, right? And then vascular inflammation also happens, right? And these are, I mean, if you can imagine, these are sort of big calcium sort of deposits in our big arteries. So they start with big arteries and then they can go on to the smaller arteries, right? And also high TMAOs are associated with, even high levels of them are seen in myocardial infarction or so-called heart attack, right? Even CNS disorders. Now this is something interesting, right? So gut associated with CNS disorders. So gut microbiome has been associated with Parkinson's disease, Alzheimer's disease, right? Even autism as such, right? So these are all something, this is all associated, as I told you, with the leaky gut as such, right? And dysbiosis sort of happens, right? GI permeability, lot of toxins, metabolites are accumulating in the system. And as I told you, because there is a gut and brain axis, a lot of these signals are reaching the brain and leading to these situations. Now, eye itself has its own microbiome. Remember, anything which is exposed to the environment will have its own microbiome. So, eye does have its own microbiome. However, even the gut microbiome is associated with certain eye conditions like keratitis, right? Sjogren syndrome, that's the dry eye situation, uveitis, etc., right? Now this is something very important for a surgeon like me, right? So whenever we used to operate or we did operation, even we do today, so the most important thing to say is, okay, there are few factors which decide that when I join two parts of the intestine, does it heal well or does it leak? Leak means it doesn't heal well and the contents of from the intestine come out. So that's called an anastomotic leak. So one is the patient factors, that is how healthy or sick he is, their albumin levels, jaundice levels, whether they have been on steroids, etc., etc., any other medication, they have received radiotherapy, chemotherapy, etc. The next part, which is most important, is the surgical part, right? Whether the gut was healthy, whether it was sutured well, and the surgeon could decide that this is the healthy part, unhealthy part, right? And then anastomosis. Now there is something new which has come up, right? So it has been shown that a higher content of Pseudomonas aeruginosa, for example, if it has been put in rats, right? So whatever your level of anastomosis, quality of surgery, etc., those having a higher content of these bacteria do have a higher associated with anastomotic leaks, right? So this is something totally new. Now, do drugs interact and change the gut microbiome? Yes. So the most important that is happen is proton pump inhibitors, right? That is whatever you take yourself as antacid, pantoprazole, omeprazole, etc. So what does it do? So once you take all these PPIs, what we call as proton pump inhibitors, so it changes the acidic content in the stomach. So what happens? Our oral microbiome, which is there, can now go into the gut. So the microbiome structure in our gut changes, all right, and then leads on to so many sort of things, right? And these, when the gut changes, so when you take drugs, for example, metformin is something taken for diabetes acid, right? So it changes the gut microbiome. So there is short chain fasciitis producer which changes how the drug is efficacious in our body, right? Another antiviral drug, right? Brivudine, right? It is converted to another metabolites, right? And then it can lead to toxicity in the gut. So levodopa is another one, right? Which is converted to dopamine as such. So, of course, antibiotics do change our gut microbiome, no doubt about that, because that is the job of an antibiotic. Killing, right? Killing or whatever of the bacteria. And because of all this, right? So, drugs and gut microbiome, when they play around, right? So they can transform drug structure and then they can change bioavailability, bioactivity, toxicity as such. This is another interesting part. So it's very common. So nowadays, so what is NAFLD? NASH. So NAFLD is non-alcoholic fatty liver disease, and then if it progresses, non-alcoholic steatohepatitis. So what is NAFLD? So NAFLD is basically deposition of fat within your liver. So normally you have a healthy liver, less than 10% of our hepatocytes will have fat in them. But once you have more amount of fat in your liver, right? And then what happens is if you have more amount of fat in your liver and it progresses, it leads to inflammation. So then it can very well lead to, like you have cirrhosis, you take alcohol leading to cirrhosis. Cirrhosis means the liver becomes firm and nodular. So very similar thing happens. Just because of high amounts of fat in our liver, you can your liver over a period of time, right, becomes hard, cirrhosis, leading to even whether malignancy formation like hepatocellular carcinoma or even needing liver transplantation as such, right? So this is again very similar. So when you have dysbiosis, high fatty diet, alcohol, low fibers, or X factor, whatever it may be, right? And the gut barrier changes, and there are volatile organic compounds and pathogen associated PAMPs, etc., right? Which again lead to this NAFLD and NASH as an entity. Microbiome again leads to encephalopathy, right? So when we have dysbiosis, especially in the setting of a cirrhosis, right? So we have amino acids, tryptophan, they are generally produce ammonia and indoles, right? And the indoles change over to oxindoles, right? But our liver in a healthy situation is able to detoxify this. But in the presence of cirrhosis, when these are produced in higher amounts, they actually cross the blood-brain barrier and go into our brain. And that is what we call encephalopathy, hepatic encephalopathy, leading to coma as such situation. CA colon is very something which is sort of proven, as I told you before, right? CA colon, high red meat content, high fat content, low fiber content, leads to change in the gut microbiome, right? And then leading to colon cancer, right? And then now they have even shown that even recurrence of colon cancer, so we have resected them, we have removed the cancer, the chance for it to come back is higher, right, based on our microbiome as such, right? So this is a very nice graph which I just picked up just 2 days ago from a conference that I attended, right? So it very clearly shows the lower red line, right? So if you see before surgery, both of them will have very similar bacteroides picture, and then intra-op only it changes because the bowel prep, that is the preparation that we give to the bowel, is different. And this is very important. If you see the blue line, what's happening there is, this is intra picked up. So what here has happened is, when we do a colonoscopy, we only give bowel preparation. So it doesn't change much. But when we do surgery, we give antibiotics, intraluminal, that is inside. So you can see the moment you give antibiotics, their microbiome has changed, bacteroides, right? And then about 1 month after surgery, they are coming closer to what they were previously, right? Especially post surgery. Now you have to remember that antibiotics, as I showed you here, changes our gut microbiome, but there are certain antibiotic related situations where your gut microbiome may never come back to normal as such, right? So now, learning all this, now we should look at what are the treatment or therapies that we could have as such, right? So these are certain terms that better for you to remember, that is probiotic. So what's a probiotic, right? It's a live microorganism that when administered in adequate amount confers a health benefit on the host. So the simplest probiotic that you have every day is curds or yogurt, right? That's a probiotic, right? Prebiotic is something which is a fermented ingredient, right? That will have, because of its changes that happen, right, will confer benefit to the individual. And what we use in our practice is lactulose, lactose, right? These are sort of sugar-based things that are used even in hepatic encephalopathy. So that is a prebiotic. What are symbiotics? Symbiotics is then a combination of both, right? And then you have fermentations, right? So when microorganisms change food into other product as such. Now, if you look at therapy in general, you can have probiotics, right? As I told you, commonest thing is the yogurt and the curds that you use, they have a lot of, right, strains in them, which helps you, right? Whether different lactobacillus or bifidobacterium, etc., right? Thermophilus, etc. Prebiotics are again these are non-digestible carbohydrates, oligosaccharides or polysaccharides, which again studied. And the last is the fecal microbiota transplantation. So, I mean, I told you that NAFLD prebiotic therapy helps in decrease of lipogenesis and reducing of fat. So, in general, probiotics, they have immunological benefits as well as non-immunological benefits, helping us as such. Now, when we come to clinical practice, right? So these are all, many of them are laboratory studies, associations in different animals that has been studies, right? But when I want in my clinical or in my practice, right, I want to prove something that it is beyond doubt. So it's nowadays what we practice is called evidence-based medicine, right? So I want to have proof beyond doubt that this actually works. So when we look at levels of evidence, right? So let's start from number 5. So number 5 is, I may tell you something, right? The professor just says, I think this works better, so this should be done, right? So it's a level 5. It's an opinion of an individual. They may have, of course, based that opinion based on their experience over a period of time. But if it is not been proved in a setup, right, it has low evidence level. Then when you go to level 4, that is when you do a study based on number of cases, right, or based on historically controlled studies, that is level 4. Then you have a non-randomized control cohort or follow-up study. So this is a non-randomized study. So right, you just picked up a bunch of individuals and studied them, right? So they are level 3 evidence. Level 2 is when you are randomized. So randomized means, so for example, I decide I will look at whoever has surgery, whether they will get infection or not infection, and associated with some therapy. So I will randomly divide them into 2 groups based on computers, right? And once I randomize them, then I do my study, and because I have had no bias, and there are levels of different randomized blind study where the investigator has no idea who is in which group, and they go through. That is level 2 evidence. And level 1 is when a number of randomized studies are taken and then you review them or you do a meta-analysis of these number of randomized studies and come up with an answer. So that is level 1 answer. Now I have picked this up from the World Gastroenterology Organization, their guidelines. Right? So believe me, we talk so much of so many things, but in clinical practice, presently, we are very, very much limited to what we use in our day-to-day practice. So if you can look at this, so look at the evidence level on the right side, right? And then look at this column, right? So if you have a number 1 there, it is very good, right? So as it number increases, the evidence level is lower. Remember, level 1 is the highest, level 5 is the lowest. So where do we use it? Antibiotic associated diarrhea. Yes, right? So we have lactobacillus, streptococcus, right? Bifidobacterium, etc. Right? So this we do use in practice, right? Now, acute diarrhea, if some adult just has an acute diarrhea, should you use or not, right? But the evidence level is quite low as such here. level of 2 and 3, right? Now H. pylori eradication, again the evidence level is 2 here, so you could use it, right? It is possible. Hepatic encephalopathy, I told you about, right? Hepatic encephalopathy with in the presence of a cirrhosis. Yes, I told you, right? So lactulose, if you see there, right? Level 1 evidence, use of lactulose, right? And we do use it in our practice regularly. So patient with cirrhosis, hepatic encephalopathy, or in a similar situation, we will use this lactulose as such. But again, yes, level 2 is also good enough for us to use in our practice. So even level 2, for example, evidences, these are a mixture of different strains of bacteria that we use. Irritable bowel syndrome, not so good evidence as such, right? Constipation, if somebody has constipation in your family and says, should we take a lot of all these probiotics? Not so good evidence as such, right? Now this is something interesting that is there. Post-operative sepsis in elective GI surgery, right? So they have given them a lot of pro and biotics, etc. And they have found significant benefits in improvements of this sepsis. All this again, so in IBD remission is when there is something known as the inflammatory bowel disease, whether it is ulcerative colitis or Crohn's disease. So one is when it is flared up, so when there is high activity, and the next is when it is remission, it is in low level. So in remission, yes, it does benefit, you can use it, right? Also, many of you don't like to take milk, probably lactose intolerant, etc. So in lactose maldigestion, it does help, right? Taking the lactobacillus, particular strain of it, which helps it. Acute gastroenteritis. So you had a pani puri, masal puri, etc. Right? You went and had, and then the following day you had a bad diarrhea out of it, right? So yes, it does help. So Saccharomyces boulardii, etc. Right? It does benefit you in that situation. Correcting your gut microbiome would be helpful, right? Now this is again very important, antibiotic associated diarrhea. So I mean, I don't know if whenever you would have taken antibiotics for whatever infection that you had, you might have seen that your gut sort of keeps changing. You may have a little bit of constipation, gut feels bloated, etc. So yes, when you have antibiotic associated diarrhea, there is strong evidence that it does help in the treatment. Infantile colic, small children having colic, etc. Colic is pain as such. Yes, there is some evidence that it does help them as well. Now coming nearly to the end of my talk, this is fecal microbial transplantation. So remember we talked about poop soup. So believe me, this is what is done, and this is FDA, right? This is from the US FDA. So presently it is approved only for Clostridium difficile infections, right? Something known as pseudomembranous colitis that develops in us. So what is done is from healthy donors, poop is collected, right? And this is analyzed and prepared. Then it is implanted into the gut of the recipient or the individual who is sick, right? And this could be generally placed through any forms of tubes into the GI tract or either through endoscopy, so you can place this quite lower down, or colonoscopy directly into the large intestine, right? So presently, fecal microbial transplantation is a process that is used, right? And presently approved only from Clostridium difficile. However, there have been cases of inflammatory bowel disease and other chronic diarrhea where it is being tried out and hopefully will be successful. So to summarize my talk, right? So as I showed you, there is an explosion of publications on gut microbiome. Means there is a lot of interest that is happening. And gut microbiome is practically associated with disorders with every organ system in the body, right? And there is a major scope of further study, as presently it is all associations, as I told you, right? But we need further proof of only that being the only causative factor. And as I showed you, there is a therapeutic potential, but I do believe that it will have a huge therapeutic potential in different formats over time. Thank you.
Thank you, sir, for that enlightening talk about the microbiology of the gut. There was so much to take away from that, and I'm sure that everyone here today will agree with me when I say that it was a pleasure to listen to you. I'm sure that there are a lot of you itching to ask questions. So without further ado, let us begin with them. The first question is by Arun Kumar E. Can fecal transplant from an obese to lean individual lead to weight gain? When a person dies, what happens to their gut microbiome? Can the time of death be predicted?
So what has been happened is they have done mice studies and they have shown that if I pick up the gut microbiome or the gut contents from a fat mice, right, and then transplanted it into a lean mice, yes, they tend to become more obese. So yes, you can transfer things like that.
Continuing from that,
Yeah.
When a person dies, what happens to their gut microbiome? Can the time of death be predicted for forensic studies?
Presently not. I have not seen any study which can use it in forensic medicine as such, right? But in the future, I have no idea.
Okay. Going to the second question by Vaibhavi Kulkarni. Are food intolerances linked to the gut microbiome?
Okay, so what has happened is, one truly is lactose intolerant, right? So I showed you that people who are lactose intolerant, right, cannot digest lactose and they may not like to drink milk. But when you provide those lactobacillus, particular strains of lactobacillus, their lactose intolerance disappears. So which means it is absence or reduction in a number of that particular gut microbiome, which will change your ability to digest lactose as such. Yes, but nothing of such has been proved in other conditions.
Shlaga Sharma asks, I have read somewhere that there is a class of certain bacteria whose deficiency can lead to stress and anxiety. Can you throw some light on it?
Yes, so this is something known as an irritable bowel syndrome as such, and a function. These are all comes in the category of functional disorders, right? So functional disorders mean you cannot pick up something anatomical, pathological, etc., right? And we do very well know that there is a gut and brain axis, as I showed you very well, right? So the more the leaky gut, and then more of the metabolites, etc., are going to your brain as such, right? And they lead to different situations. Yes, there is association between your health of the gut and depression, anxiety to an extent. Yes.
From Ms. Shalmali Kamath, gut microbes are known to produce serotonin. So does gut microbiota influence or control our mood or psychological state? Can the gut microbiota have an effect on the intellect of an individual?
Intellect, I have not I try to look at, I don't think there is anything on that, right? But yes, as I told you, mood will differ, depression, right? These are the things which does change based on your gut microbiome.
Another question from Arun Kumar E. How does our gut microbiome respond to fasting? Does high ketone level affect bacterial gut population during fasting?
Okay, I think this is something which is interesting in terms of recent your keto diets as such, right? So you have a lot of ketones, you use a lot of proteins, mostly protein diet and fat-based diet rather than carbohydrate diets, right? So your ketone levels goes up, right? So it does change your gut microbiota, right? And as I told you, you start smelling of those ketones as such, and it is again related to your kidney injury as such, right? And there is something which is very interesting again, right? So there is something known as immunosenescence, right? So as you age, right, your adaptive and innate immunology, right, changes over a period of time as well, right? So what it does is your immune ability to fight a disease gets lesser and lesser as you age over time. So this has also to an extent been associated with the gut microbiome.
Moving on to the next question by Isri Inamdar. How does the gut microbiome remain unaffected by the digestive enzymes which are present around them all the time?
Okay, so I think there are two components to the gut microbiome, right? And then when we say enzymes, that is why you will see that you have very little microbiome in the stomach because it is a very highly acidic sort of environment, right? But when you come down, so once you move into the duodenum, you have bile juices coming in and pancreatic juices. So these bile and pancreatic juices change the environment to an alkaline situation, right? So these alkaline situation, they have no problem to stay alive as such, right? And then when you come to the colon, it becomes a more anaerobic sort of situation where many of these gut microbiome will thrive because of just the position of their placement.
The next question is from Ms. Riha Shah. Could our microbiome hold the key to understanding the onset, progression, and perhaps cure to cancers?
So these are all sort of under investigation, right? As I told you, presently there are situations where we say this particular microbiome is associated. Now, one is obviously proven, H. pylori is associated or proven to cause cancer stomach. So there is no doubt about that, right? So this H. pylori is also a gut microbiome. So to answer, yes, gut microbiome has been proven to cause cancer, but for other cancers, as I told you, for example, CA colon as such, right? It's an association rather than a direct causation. For example, I cannot tell you like Mycobacterium tuberculosis causes TB. So it's not a direct association there because there are so many other factors which come into play and so many metabolites, proteins, etc., which are produced and they produce so many different effects. And eventually, because of other environmental, other genetic causes that you will develop cancer. So this is an additive sort of factor.
Moving on. Another question from Arun Kumar E. Does quorum sensing contribute to the microbial balance in the gut more than the diet?
Sorry, can I get the question again?
Does quorum sensing contribute to microbial balance in the gut more than the diet?
Honestly, I have no answer to this.
Okay.
I should
Moving
look
on.
it up.
Okay. The next question. What is your opinion on the Indian gut project? Why hasn't India focused much on the gut project like the American counterpart? Again by Arun Kumar E.
So I think when we look at Indian scenarios, whether it is the government or non-governmental organizations and all, we have so many other priorities which become much more important, right? So we have infectious disease sort of is the main priority, right? And now we are going to non-infectious diseases, right? So whether it is cancer or hypertension or diabetes or kidney disease, etc. So it is just a question of the priority and the resources, right? So obviously, if you have the resources, you can do any sort of project. There is no hindrance.
The next question is from Dr. Syed Wajid. For how long does transplanted gut flora remain? Again, there might be dysregulation because of the existing microbiota overpopulating, and the environment may also not be conducive for proliferation of the transplanted ones.
You are very right in that idea there. So the transplanted gut microbiota have been studied for short period of time presently, 2 to 3 years, right? And they seem to be present there for a decent amount of time. But the advantage you have is, right? So what you can do is you can have repeated sessions of transplantation because it is a very minimally invasive procedure, right? So you have a nice stock of these bacteria, right? Repeat this transplantation process as many times as you need. So you can repopulate your gut microbiome with the healthy microbes.
The next question is from Monica Pradeep. Does the microbiome play a role in absorption of nutrients in our body?
Yes, I told you, remember, that when you compare an obese individual with a lean individual, the microbes in the obese individual pick up more of the calories which are coming into the gut than that of a lean individual. So yes, microbes do pick up more of these calories and sugars and things like that.
The final question is from Dr. Beatrice Sequeira. So, would there be personalized fecal microbial transplants as we have diverse microorganisms and geographical areas?
I think, as I told you, presently fecal microbial transplantation is limited for a particular disease, Clostridium difficile or pseudomembranous colitis, right? Presently. But it is being tried in inflammatory bowel disease, irritable bowel syndromes, chronic diarrheas, etc. Right? But presently it is not of so much success. So the regulatory bodies allow it only for Clostridium difficile infection as of yet, right? But obviously, once we understand which are those bacteria or flora which are of more benefit to the individual, then only we can go ahead with that, right? And this is an evolving field, so I'm sure it will happen over next 5 years or something that you will have sort of personalized, giving probiotics, right, in a personalized format. So an individual comes up, we do a genome analysis, metagenome analysis, and say these are your bacteria, right? this is more healthy, this is less healthy, or this you have more in number, or this is less in number, but I want to increase this less number to the higher number. So I may have a concoction of different, bacteria to individual basis. So that is the goal, yes. In fact, we are also working towards, with Professor Tatu at IIC, towards that goal. In fact, we are doing 2 studies at the moment on gut microbiome. One is in the presence of chronic pancreatitis, right? How it matters. Second thing is gut microbiome post surgery. So we do something known as hepaticojejunostomy, right? So we put in the jejunum to the bile, right? So the intrahepatic circulation system all changes. So it has been seen that in bariatric surgery, one is just because of the structure, then because of the microbiome changes, you also lose weight. So we actually want to study what are these, what is the changes that actually happen in gut microbiome, and which are the phyla or which are the species that increase in number and which decrease in number.
Thank you for the questions, audience. Thank you, sir, for answering them so patiently. Your questions, I'm sure. Thank you. Your questions, I'm sure, helped all of us to see more perspectives in today's talk and learn more because of it. I thank both our illustrious speakers for being here today and making time to give us wonderful talks that have broadened our horizons. Thank you all again for the insightful questions. I will be taking your leave now. Over to Calderda.
Thank you.
Thank you, Sumeru. And with that, we have come to the end of the events for day 1. It has indeed been a stimulating experience for our minds listening to the lectures of eminent resource persons. Please note, all participants can join back for the lectures tomorrow using the same ID and code. Thank you all for joining us today. We hope to see you tomorrow morning at 9 a.m. for day 2 of the lecture workshop. Thank you. Have a pleasant evening.
Thank you.
Thank you, sir.
Thank you, Dr. Rao.
Thank you.